Good morning, everyone. Thank you for joining the webinar. This is Joshua Xu. I am from FDA. I am the Executive Secretary of the MAQC Society. It is my great honor and to give you a short introduction of our society and today's speaker. So MAQC Society stands for Massive Data Analysis and QC Control Society. And we, from the name, you can see that we focus on two things: massive data analysis and reproducibility. And we examine the existing and emerging technology and how those technology can be used to advance our understanding of biology and healthcare. So for this webinar series, for some of you who have known that we have hosted a series of webinars for FDA-led sequencing quality control phase 2. So for today and the next 2 weeks, and we're going to have webinars for the working group, Oncopanel sequencing working group. So for today, that Dr. Wendell Jones we're going to present that how the working group developed a reference sample to enable such analysis and the evaluation of Oncopanels. So Dr. Wendell Jones is consulting statistician and bioinformatician in genomics technology and systems, serving in leadership roles with an FDA-led consortium such as Sequencing Quality Control, SCQC, and SQC2 Consortium. And it is my great honor to introduce him because not just that he is the current president and the executive chair of the MAQC Society, and he also plays a tremendous role in the Oncopanel sequencing Oncopanel sequencing working group. So it's Dr. Jones is like my mentor for the working group. So at EA Genomics, Dr. Jones was Vice President of Statistics and Bioinformatics, Global Head of Bioinformatics and Clinical Systems, and he is currently Principal Bioinformaticist and Scientific Advisor. Wendell, the floor is yours.
Thank you, Joshua. So yes, it is a great honor and privilege for me to be able to present this information on a new verified genomic reference sample for assessing performance of cancer panels, looking at small variants, meaning SNVs and small indels of low allele frequency. And let's go ahead and begin. So there's a challenge with developing and validating cancer panels. The challenges are things like somatic mutations are observed at low allele frequency, usually less than 20% allele frequency. And this can be due to a variety of reasons. For example, you may have low tumor purity. There could be variants that are under copy number influence. There can also be heterogeneity in the tumor, and you may have an emerging clone that has a driver variant at low allele frequency. And of course, there's applications in liquid biopsies where plasma is collected and cell-free DNA is extracted from that, and you would often have variants at very low frequency there because you're looking at the dying cancer cells versus the background of normal cells also dying. So when it comes to Oncopanels and liquid biopsy panels, one challenge is they're often interrogating either tens of thousands to tens of millions of genomic positions for this kind of variation. And so when examining that, there's a question of how do I know that I have validated sufficiently the regions that I would like to interrogate. Related to that, many established reference samples, whether they're commercially available or internally developed, they usually only have a relatively few low variant frequency positives that are identified, for example, in coding regions or even overall. And these usually only encompass a relatively small number of genes and positions. The other challenge is that if you do develop a reference sample yourself using actual tumor samples, One challenge there is that materials of limited duration, because typically you're either through QC or through development or further validation, you have to continually use that material. And since each time you use the material, you use it up, then at some point you will run out of that material. So it's not long-term or perpetual. Another challenge is, how do I establish things like a valid limit of detection with only a few variants having frequency around the true limit of detection? So if the limit of detection for your panel is 5% or 2% or below 1%, how many variants are you testing in that range? And if, even if you find all of them, is that really establishing your limit of detection? What if you only find, 2 out of 4? How do I infer what my limit of detection is? Just in general, how to determine sensitivity and precision and other key metrics with reasonable confidence bounds with only a few variants in a particular reference material. So these are examples that I've encountered personally myself working at a laboratory. So we often get questions from our assay development team, for example, on, let's say we have a new Oncopanel, it's only looking at relatively few regions, and we have a reference sample that may have 8 variants that are documented between 5% and 12% very low frequency. So I run the sample and I detect, say, 7 of them, 7 of the 8. What can I infer? What if I detected all 8? What if I only detected 5 of the 8? And the answer is, I really can't infer much, statistically speaking. We don't have any replicates, so there's no inference about the 8 positions themselves. And our sample size for general inference about all positions is just really small. Okay, so what if I have an Oncopanel that I run 5 times with some verified reference sample that has more variants? Let's say it has 20 variants around a 5% very low frequency. And let's say I detect 20 out of 20 each time. So essentially I get 100 out of 100. That's pretty good, right? What's my confidence level, my confidence interval for the sensitivity? And also, can I treat the different runs as independent observations? And the answer is, making inferences about a specific genomic position is different than inferring about all the positions that your panel is testing. With replicates on those 20 positions, those 20 variants, you now can make turns out a rather poor inference about these 20 positions, even if you find all of them. Turns out the 95% confidence interval on the true rate of detection is only, I should say, is so wide, 40% to 100%, that it really doesn't tell you much information about the ongoing reliability or the sensitivity of your assay. And the sample size of 100, you really don't have a sample size of 100 because the replicate observations are heavily correlated. Given the fact that you detect it once at a given position, implies that you're very, very highly likely to detect it again at the same position. It doesn't offer additional inference about other positions. So just because you detect 100 out of 100, if they're done through replicates, it really doesn't generalize well. Another example, let's suppose I have a small Oncopanel and I have a reference sample with 15 variants that seems to work well as one of the key validation and QC samples, and that I've run this particular reference sample many, many times to get a much tighter confidence interval. But now I'm testing a much larger panel, let's say bigger than a megabase. So, and I do that so maybe I can estimate TMB or some other important endpoint. The question is, can I infer sensitivity for the much larger panel? Is it the same as the smaller panel? Or do I need to find a different reference sample? And the answer is most likely, as the kits may have very, very different properties outside the limited regions in the small panel. So you can't take the results from a small panel and just automatically assume that you'll get the same results for the much larger panel. Well, these were some of the challenges that we had in the Oncopanel working group. Was we wanted to perform an extensive performance evaluation of current and developing Oncopanels that have varying sizes, as well as liquid biopsy panels. We wanted to understand what's their general sensitivity and precision, what's their limit of detection, can some panels be used to estimate mutational burden, are false positives under control. We examined existing commercial samples, and they typically had very few variants, whether hotspot positions or not, at low frequency. And when we looked at NSGM and Avadel samples as is, they weren't suitable for somatic analysis because they're more or less identifying germline positions, usually variants at 50% allele frequency or higher. Now we had a partner working group 1, that did tremendous work on a breast cancer normal cell line, the HCC 1395, where they were also trying to identify variants, but they were looking at it whole genome sequencing primarily. And they would document millions of variants and be able to evaluate different whole genome sequencing and whole exome sequencing platforms from that. But for our purposes, since it had relatively few, just given that it's one breast cancer cell line, there are relatively few low frequency somatic point mutations in coding regions. In fact, less than 150 across the whole exome, which was a number that was comparable to some commercial offerings. So we decided that we needed to examine a new material, and what we decided to do was take a material that we had used before, but use the RNA portion, that is the universal human reference RNA source that had been used for 3 different instances of the consortium work in microarrays and RNA sequencing, but just look at the DNA portion. So this source is based on an admixture of 10 cancer cell lines from different tissues. We already had extensive sequencing data from RNA sequencing, and so potentially this could yield a source of material with tens of thousands of low frequency variants. So in our Oncopanel study, we actually, as Joshua mentioned, we have several components in it, and there will be more that will be discussed in the next 2 weeks about, for example, the pan-cancer study and the liquid biopsy core study. In this particular case, I'm focusing on the reference sample itself, which is the basis for how we evaluated things like the pan-cancer study and the liquid biopsy study for many, many different panels, as well as looking at things like target RNA-seq, and also examining, for example, the impact of FFP on this material. So we were using this as a basis to inform other studies in the particular working group about the performance and characteristics of these different Oncopanels and liquid biopsy tests. Okay, so let's talk a little bit about the cell lines that were used in the study. So as I mentioned before, we were using the 10 cancer cell lines that are associated with the universal human RNA reference. You see a list of those numbered here. They run from a couple of different types of blood cancers to brain cancer, breast cancer, liver cancer, etc. And there's also a normal male reference that I'll talk about in a few minutes that was used to admix with these materials so we could get variants even at much lower frequency, for example, for use in liquid biopsy. They did have, the cell lines are distinct in lots of different ways. So in addition to different tumor sources that are going to be used to admix to create our reference sample A, as we like to call it, we see that, for example, they have varying levels of copy number alterations. So some, like the liposarcoma and the brain cancer, it's almost like the vast majority of positions are under copy number influence, whereas others, for example, like the melanoma and some of the blood cancers, have relatively little copy number changes. But there's also things like, even though it's a cell line, it may be polyclonal. And in fact, we identified that several of these cell lines appear to be polyclonal, with the breast cancer being maybe the most extreme in terms of being polyclonal. And so we had to consider this when examining what we could infer from individual cell lines leading into the pool sample A. So the strategy was for discovery of the variants in this sample A material, which is a mixture again of these 10 cancer cell lines, we decided to enlist the help of several different whole exome sequencing enrichment panels, and we numbered them 1 through 4. So that includes number 1, the Roche MedExome panel, again, a whole exome panel that was used to interrogate the individual cell lines that make up what eventually would be the pooled sample A. For the Roche MedExome panel, for the IDT XGen exome panel, and for the Agilent SureSelect exome panel, we decided to run these at very high depth because we are looking at both cancer cell lines and ones that may be polyclonal. may have other interesting properties like under copy number influence, as I mentioned before. In fact, one of them, the T lymphoblast cell line, has an additional interesting characteristic in that it is inherently polyploid or tetraploid because it's a fusion of a normal and a cancer cell line. And so we would run 2 libraries of those. And then for additional verification, we also use the Thermo Fisher AmpliSeq exome panel, where only 1 library was created. But the idea was to run the individual cell lines with each of these panels to elicit the variants that were in the individual cell lines, which would eventually be in the pooled sample A. We also ran whole genome sequencing using the 10x genomic linked read prep. There we only did 1 library per sample, and was also used to discover variants, and I'll talk about the rules for discovery in just a second. Given all this sequencing that we did, we then had to decide what genomic regions will we infer have variants, that is, are positives, and what regions will we identify as lacking variants, because it turns out identifying regions that maybe do not have variants could also be very, very important, especially when you're looking at things like precision and false positive rates and things of that nature. So let's talk about that for just a minute. Related to the whole exome sequencing kits, we decided that we would focus on the regions where each of these kits overlapped in terms of their target regions. So again, we have each whole exome sequencing kit 1 through 4. We identify, given their target regions and their bed files, what the overlap is, and we called that set of loci the interval 4 set. We took that interval 4 set and then we overlapped it with the human coding regions. And the reason this is important is, as you may know, each of these whole exome sequencing panels, while they do interrogate the whole exome, they also interrogate regions overlapping or outside of the exome, say, that are overlapping the gene, but not necessarily in the coding region. They could be in UTR or in introns. We wanted to focus on just the coding region. So we took the intersection of this interval 4 set with the human coding region. And then early on, we made a discovery that we could be much more certain of our results if we also examined the NIST high confidence regions, or sometimes called the NIST benchmark regions, based upon some genome in a bottle work that was done by NIST. So we intersected the interval 4 coding regions with these NIST high confidence regions, and then we did a small exclusion of some additional low complexity regions. And that became what we entitled our consensus target region. And in terms of the size of this consensus target region, it's still pretty substantial. It's about 2/3 of the entire exome. And if you look at the interval 4 coding region, which is about 80% of the exome, and then the high confidence regions, which is also about 80% of the exome, it turns out it's pretty much like an independent overlap of those two. If you do the multiplication of 80% times 82%, it comes out very, very close to 67%. The other condition we made is, just for simplicity, we decided to look at autosomal regions only. Okay, so we've decided what regions we want to call variants in. And so now the question is, with what methods will we actually identify the variants? And initially we examined lots of different variant calling pipelines. Let's talk about that in just a minute. Even though we started out with a much larger set, we focused on a little bit more than 20 of the final pipelines that were implemented across several independent institutions. And you see the list here. And as you can see, if you look through the list, a lot of them are with things like MUTEC, versions 1 and 2, things like VARDICT, things like SureCall and VARSCAN. Really what we did was we wanted each institution to use a pipeline with which they were very familiar and had reasonable to high confidence in. And we thought that was important because there are still a lot of parameters that can be tweaked with these different pipelines. We wanted individuals who are running them to have confidence to be able to justify and defend the results they were getting, to make sure they were of good quality. So this was the initial set of both mappers and callers that were used to identify potential variants both in HG19 and HT38. And then we had to determine rules, the final rules for saying, okay, which variants that come out of these pipelines are we going to say are positives? And the final rule that we came up with by acclamation, because it was difficult to say, given the patterns of the variant calling, that one caller would naturally be, let's say, favored over the other, we decided that by cell line, that if a variant was called by at least 50% of the pipeline library combinations in each of the three high-depth whole exome sequencing systems, then that would be a candidate for a positive. But then it had to have some other characteristics too. So for example, it had to be a very low frequency in the cell line that was greater than 10%, and the variant had to be biallelic. And then in addition, it had to be, for example, for a class 1 variant, it had to be in the consensus target region that I just defined. However, we noticed that there were some maybe important regions that were not in the consensus target region, yet were still in the high confidence exome. And so that's where we brought in the whole genome sequencing results. So for the class 2 variants, our rule was amended to say, okay, it has to be called by at least 50% of the pipeline library combinations in at least 2 of the kits, but then it also has to be called by both callers that we used for the linked read whole genome sequencing, and also obviously not be class 1. So basically, if you were found in all the kits, or if you were found in 2 of the kits because maybe one of the kits wasn't covering a particular region, but it was also found by whole genome sequencing, which by definition would cover everything, then it could also be a positive variant. We just identified them as class 1 versus class 2. Of course, there are going to be many more class 1 than class 2 because there's a lot of overlap, between the different kits. From that, we were able to identify over 42,000 consensus variants in high confidence exome regions, and that included variants in more than 2,400 different cosmic genes, cosmic being the catalog of somatic mutations in cancer. And the majority in each of those groups of those variants have a very low frequency, less than 20%, in the pooled sample A, but not necessarily in the individual cell line. So let's just illustrate that a little bit more fundamentally, taking a archetypal gene like P53, just to illustrate, for example, that the kits that are used to enrich for the exome are a little bit different. You see tracks for how each of them interrogate this particular gene. They're very similar, but you also notice the differences between them. You also notice that the interval 4 region is going to be the overlap of all 4. So you can't have a base in the interval 4 region unless it's covered by all 4 of the kits. Then you have the NIST high confidence regions. You have the exclusion of the low complexity regions, and that gives you the CTR, the consensus target region. And if you look at our known positives that we identified ultimately for sample A, that is the pooled sample, you see that we've identified 7 variants in this gene in the sample A material, and they're both class 1 and class 2. The class 1 genes are not, I shouldn't say encircled, but they're not put in a box. The one class 2 variant, which I have identified here, why is it class 2? Well, the main reason is if you look at everything else. So for example, it's not in the CTR. It's also not excluded by a low complexity region. It is high confidence. But we look at it, turns out that the adjuvant sure select is just not covering that part of the exon. So the other 2 are, and it's detected there. It's also detected in whole genome sequencing. Therefore, that's what makes it a class 2 variant versus a class 1 variant. All the other variants are in the CTR. When you break down the total set of variants that we identified for sample A, you see that, again, it's a very, very large number, 42,000 in total, 28,000 of them being allele frequency less than 20%, which is really tremendous. That gives you a lot of power and sampling power to understand how well your methods and pipelines are doing throughout the exome, at least in the high confidence regions. Also, if you look at genes that matter, like cosmic related genes, you see that we have 1,600 variants we've identified in cosmic genes that have an allele frequency less than 20%, which again is tremendous. And you see how they're broken down into class 1 and class 2. For class 2, we decided to focus a lot on the cosmic genes. So whereas class 2 variants don't take up a large number of variants overall, they do comprise more than 10% of all variants in cosmic genes. So again, even though they're less than 1% of variants overall, if you look at cosmic genes, they're more than 10% of all the variants we identified in cosmic genes. Now, if you look at sample A compared to, for example, some other efforts and some commercial samples, some samples that are very, very good. So Acrometrics provides a reference sample that has over 555 variants, 341 of them are below 20% allele frequency, although in one sense you can actually work with Acrometrics and get all of them at a lower frequency if desired. Oncospan is another good reference material that has a large number of variants. But again, if you look at them compared to what is available in sample A, you see that sample A often has, up to 100x more variants of different types. If you look at our sister effort in the whole genome sequencing working group, you see that they identified a tremendous number of both germline and somatic variants. In fact, their total somatic variants in HCC 1395 are comparable to what we identified. But again, that's across the entire genome. If you look at how many of those variants are actually in coding regions, you find out it's much less, about maybe 144 that are, for example, in coding regions that have allele frequency less than 20%, and only 14 of those are in cosmic genes. So again, a great reference material, especially for whole genome and whole exome sequencing in general. But in terms of utility to having a very, very large number of low frequency variants in coding regions, we decided that we needed something in addition. All right, so we've identified these variants, we understand their characteristics, and now the question is, can we confirm them, and what would be a way of confirming them? So the main characteristic that we use to confirm them is actually interrogate the pooled sample A itself. So as we mentioned before, these variants were identified by looking at the individual cell lines. And so now we want to confirm them by looking at sample A itself. We'll do that in two ways. First, we're going to actually create the pooled sample A, and then we're going to sequence it to very high depth using the three kits that I mentioned before, the Roche panel, the IDT panel, the Agilent panel. We're going to sequence them in triplicate each to very high depth, and we're going to merge the BAMs from those triplicates and then call variants from that merged BAM for each of WS1, 2, and 3. We're also going to look at an in silico merging of individual cell line BAMs by kit for both HG19 and HG38. And then we're going to call variants from that in silico merging as well with a certain select pipelines. And when we do that, we find that, in fact, 99.7% of all those 42,000 variants that we detected with the individual cell lines were detected in at least one kit of either the merged library BAM sample A or the in silico sample A using only one variant calling pipeline. In this case, we use SomaticSeq because it's an open source, readily available pipeline that anyone can find. But not just that, 96% of them were found in every whole exome sequencing kit that we use for the merged sample A, as well as being detected in all the instances of the in silico A. So both, basically 96% were seen in all the kits, both in the merged library sample A and in the in silico sample A. And then you see that some other percentages were, for example, detected in all the merged library BAMs, but maybe only 2 of the 3 of the in silico A, and so forth and so on. But again, essentially 99.7% being detected in at least one or the other is still very, very good. So we felt very, very good and very high confidence about the variants that we found, and we felt like we had confirmed that we had in fact, you would see these variants in sample A if you had sufficient depth and the other proper parameters that you need in order to sequence a sample well. Let's talk about the allele frequencies themselves, because in addition to confirming that you detect them, do you get consistent views of the allele frequencies? Because eventually you're wanting to bend the allele frequencies into different size, different ranges, I guess maybe would be a better term, so that you can understand how well your particular method or panel or pipeline is doing in allele frequency ranges, say from 10 to 20% versus 5 to 10% versus below 5% allele frequency. And so the question is, when we look at these different kits and different methods in silico and the merge library, do we get consistent allele frequency estimates? And the answer in general is overwhelmingly yes. And in fact, if you look at one particular example, where I'm going to take the kit where we had the most depth, which in this case was the Agilent SureSelect. If you look at the Agilent SureSelect merge library versus the in silico individual cell lines combined very low frequency, you see a really tight correlation 0.999 essentially. And so that lets us know that we're getting consistent results, for example, between the merge library version and the in silico version. So we feel like we're confident in using either. But even if you look across kits, you see very high correlation, usually at least 0.99 between one kit and another. And really the only time you see differences that are noticeable is when you do just a simple cell line average. And the reason that you see differences there is you do see now the impact of copy number. So copy number that may be very large in one cell line will bring that influence into the merge set. Excuse me just a second. So occasionally you will see variants that are outside the high correlation in the simple cell line average. But again, we're not going to use the simple cell line average. We're going to use the merge libraries to construct a final estimate of very low frequency, so you can be confident in the allele frequency magnitude of the variants that you're testing, so that you can bend them appropriately in estimating things like limited detection of your particular panel or bioinformatics method. So if you look at the final sample A in terms of the very low frequency distribution, you see that, again, we have thousands of variants of low frequency. In fact, you can see that over 14,000 of the variants have a allele frequency below 5%. Another 8,000 are between 5% and 10%. And if you look at the amount under 20% allele frequency, again, it's I think around 28,000 total. Just a tremendous number of variants at low frequency across the exome that you can use to validate, QC, develop new panels and test and make sure that their performance is as expected. All right, so we've confirmed them, but in one sense, what we've done in confirmation is we've used the same technology in confirming them that we use to discover them. So it's not quite a validation. So can we do, for example, an orthogonal validation of at least many of these variants that we find? And so we selected droplet digital PCR as the method to do an orthogonal validation. And for that, we designed 375 different analytes where we tested the individual cell lines. We tested sample A, we tested sample B, and we also tested some admixtures, which I'll talk about in just a minute. We did random randomization and stratification, relative randomization, to look at things like class 1 versus class 2 variants, low variant, low frequency variants, challenging indels, and variants under copy number influence. In fact, if you look at the details, this is how we broke down those 375. So we've got a set of positives, we've got a set of negatives, again, randomly selected. Within the positives, we're stratifying for, again, challenging indels, SNVs and cosmic genes, low variant, low frequency SNVs, SNVs under copy number influence, the class 2 SNVs. In fact, we interrogated almost 1/7 of all class 2 variants directly with droplet digital PCR. And then we had, again, a large number of negatives, and then we had some investigational assays as well. So again, randomization with stratification. looking at some of the important different classes of variants that we wanted to ensure were working properly. So if we look at the results for that, we see, for example, on the left, we're looking at individual cell line data. So again, we did droplet digital on individual cell line data, and we have, for example, these 284 positives, and those positives may exist in more than one cell line. So our total N in this case was over 1,000, because again, many of those variants are actually population variants in the cell line, but they can become simulated somatic in the pool. And so on average, each variant appeared maybe in 5 different cell lines. And if you look at the very low frequency estimates we get from whole exome sequencing and compare it to droplet digital PCR, again, you see a very, very high correlation between the allele frequency between whole exome sequencing and droplet digital PCR. You do see a couple of ones that are exceptions here and there. Hopefully you also see that thankfully all the negatives are at very, very low frequency. It's kind of hard to see because they all occupy one corner of the graph. But one thing you may also notice if you look hard enough is that one place where you see a little bit of discordance is in the indels. So it turns out the whole exome sequencing results will tend to show a homozygous, for example, indel as being slightly less than homozygous, whereas the droplet digital PCR will just say, no, it's 100%, this particular allele frequency, or this particular variant, is 100% of that allele frequency. And it just shows, the challenges sometimes in a nutshell that you have with indels versus other SNVs when you use short read sequencing. Anyway, if you go to sample A, and the thing here is we've switched from, say, a linear scale from 0 to 1 to a logarithmic scale. So you can see very well looking down to very, very low frequency, that what we see again is a very high correlation between the sample A on log scale and the sample A results from droplet digital PCR in the same manner. So again, and the other thing is 100% of the expected variants were detected via droplet digital PCR, and all but one of them, you can see the one exception down here, has the expected sample A variant allele frequency value given the sampling variation that occurs. I mentioned that we also did mixtures, so it turns out we mixed A with B and examined other mixtures in the droplet digital assay, that is sample C, D, and E, a 1-to-1, 1-to-4, and 1-to-24 mixture. And given these admixtures, we expect the relationship between the admixed samples and sample A to be what you see on the screen. And the question is, okay, is that what we actually saw? And the answer is yes. So if you look at sample A, which is in green here, for those variants that weren't in common with sample B, that is the sample that it was admixed with, here you see that all the variants that had that property of being independent of variants in sample B are diluting at the rate we expect. And then the sample B normal is basically showing the background, if you like, the background rate of frequency for the droplet digital PCR. So yeah, you may see higher frequency in A, but if you look at the sample B itself, we're not expecting to see variants at any frequency there. And basically you're seeing that essentially about 0.001 is the background rate for droplet digital PCR using the normal sample B. When we look at ddPCR replicates, again, we see very, very high concordance. And so that makes us think that the droplet digital PCR results are working very well. And again, sample B, the normal sample, we don't expect to see any variants, at that level, are basically at the background level and don't reproduce as expected, because again, they're just measuring background noise. So to summarize what I've just talked about and conclude, so we have reference sample A that's constructed using DNA from the 10 cancer cell lines from the Universal Human Reference RNA. It was found to have more than 42,000 variants, including 500 indels in the consensus target region, plus several other 100 variants in exonic high confidence regions. Of these, greater than 28,000 have variant allele frequency less than 20%, and 22,000 have variant allele frequency less than 10% in these exonic high confidence regions. And that's roughly 100x compared to other reference samples at low frequency. We also identified, I didn't have a chance to talk about it much, but we also identified 10 million negative positions, providing a mechanism to estimate false positive rates in the CTR. And in fact, subsequent webinars that you'll see next week and the week after will talk about the false positive rate and how the false positive rate can actually be much higher outside the CTR for analysis that we conducted with these different panels. The positives were identified using a variety of methods, whole exome, whole genome sequencing methods that were confirmed by sequencing the actual pool itself very deeply, and then a random selection of those with stratification was done via droplet digital PCR with 100% confirmation. So if we look also at the droplet digital PCR results for the admixtures, we see that the admixtures are titrating as expected, and in terms of the expected variant allele frequency, all but one had a allele frequency in the expected range given the results from sample A. So this manuscript, which has just been officially accepted by Genome Biology, will provide a lot more details than what I did here, for example, on negative positions, on the differences between the HG19 and HG38 versions. It'll even talk about the slight bias in variant allele frequency estimates using whole exome versus droplet digital. And it'll also tell you how you can construct your own reference samples. For example, how many sources of DNA do I need to admix to identify a certain number of variants at a certain variant allele frequency range? So whether or not you want to use sample A or make your own, it talks about how you do that and what you can expect those results to be given how many samples you're admixing together. In terms of reference sample A availability as well as sample B, so it turns out that Agilent made this DNA available to us in the consortium for use in both finding the variants and then testing them with the different panels that you'll see in the next few weeks. So they're preparing an alpha kit using the leftover DNA material from this effort. It should be available in the next month. And the great news is this DNA material, while it lasts, will be available for free upon request if you sign a simple NTA form at the Agilent website. And you can contact Natalia, which is one of the co-authors of our paper at Agilent, to find out more information. And they hope that this will cover customer needs until the production version of this universal human reference DNA is ready. But the sample B is already available from Agilent and can be purchased even now. And so finally, I'd like to acknowledge contributors to this effort. It was a large consortium involving 70+ co-authors and more than 30 institutions internationally. You can see, throughout the globe where they were and who they are. And so I want to give a special recognition to a few co-authors, Dr. Joshua Zhu and Dr. Benxing Gong, as well as Natalia that I mentioned previously. So Natalia and her organization provided us with the DNA, and then Joshua and Benxing were equal partners in this effort. And together we were able to manage, obviously, this very, very large consortium and get a lot of very, very good work done. I also want to acknowledge the laboratories who actually did the sequencing and sometimes the library prep for the different efforts. You see them here, as well as the data centers. We had to have some place to store all this data, and Stanford University happily allowed us to be able to store this so we could reference this data together and use it to make decisions from. And again, next week and the week after, we're going to talk more about using this information for both the pan-cancer core study and the liquid biopsy core study in the next week. So you have that to look forward to for the next webinars. And with that, I will stop.
Thank you very much, Wendell. Great presentation. And I even catch up a few things that I just really like the way that you presented. It's so clear.
Well, thank you.
So we'll open up to the audience if you have any question, and please type your question to the chat box, and so we could I could then read the question to Wendell and get his perspective. So far that I have seen comments, and so it says very exciting to have this very important resource. So it's congratulations to all of us. So while we're waiting for other people to type in the question, that I have one that, so what are the, so in your perspective, what are the limitations, and then even the maybe future effort to even to address those limitations, to advance this reference sample?
Yeah, so I think that's a great question. So there are limitations to this materials, as we discussed a couple of different times. We purposely chose to examine, in one sense, places where we felt like we could be more certain of the actual results we were seeing. No one likes to do a lot of effort, and then it comes time, let's say you're trying to do your final exams, in school. And so you do all this studying, and you hope that all the studying will yield a good result during the exam, that you'll get a good grade, and that you can move on, to the next thing. And sometimes, we get surprised and we might not get the grade we expect. So we were concerned, because we obviously did the orthogonal validation afterwards. We were concerned that, could we get into orthogonal validation and let's say only 80% of the variants validated. And so we focused on this consensus target region, which has, again, places where the different kits overlap, combined with coding, combined with high confidence regions. So what that means is that obviously there's going to be places, for example, outside high confidence, outside coding. Oftentimes there are UTRs and intronic regions that are important, for creating proteins, because sometimes changes in intronic regions may mean that splicing doesn't occur as normal. Changes in UTR regions may imply things like promoters, don't work as expected, things of that nature. So a lot of these kits will cover those different regions, but we decided to purposely not investigate those, simply because we wanted to focus on the places where we thought would have the most impact, which was coding. However, as we know, these panels will often interrogate the non-coding areas as well. So I think non-coding areas are a point of interest. I think the really difficult areas, the places outside the high confidence, things like places where you have homopolymer stretches, things where you have sequence similarity that is often the bane of the existence to aligners and to variant callers alike. And it may take efforts with more longer, saturating with longer reads and taking a reference sample to be able to identify true variants in those regions. So I think there's a lot of work that can still be done for example, almost 1/3 of the exome, even not counting the intronic and UTRs, which are probably going to be an even bigger set, versus say coding itself. But I think related to even coding itself, getting some good information about those difficult regions are going to be really important longer term for us evaluating how well different panels do in terms of especially detecting variants in very difficult to sequence regions with short read sequencing. I think Joshua, you and your colleagues Benxing will show in the next couple of weeks how the false positive rate out in those regions can be actually surprisingly high. And so, it definitely is a place where we want to examine further. And so that's, I think, a place where I would certainly welcome help in addressing.
Great, thank you. We didn't see maybe we can circle back, and we didn't see another question and posted by our colleague Wenmin. And it's a great question. And then it's obviously, and looks like that we did not distinguish germline variants versus some true somatic mutations. So if this benchmarking data set is used by the vendors to benchmarking the panel and the bioinformatics pipeline, and sometimes the pipeline would filter out germline variants. How to address this?
Yeah, that's a very good question. In fact, one of the reviewers raised a similar kind of issue. So it is the case that certain pipelines, if they are focused on somatic variants alone, may be using a reference set of germline variants in which to say, okay, if I'm going to focus on somatic variants, then if my caller, which inevitably it will, find germline variants, I want to filter those out in one sense so that I can just look at somatic. And so related to using this material in that way, that will be a setting or a change that you will need to do for your caller, because the majority of the variants in sample A are in fact germline. It's just that by putting them in the pool, of 10, now all of a sudden a variant that may be 50% in one cell line, maybe 100% in another, when you put it all together, may end up being, for example, 15% in the pool, or could be 5% or even less. But at their core, they are actually germline variants. So it is true that if your bioinformatics pipeline has already pre-filtered germline variants, either you need to account for that in your analysis and only use it for the somatic changes. And it turns out there are maybe on the order, I think we calculated maybe I would say 3,000 to 5,000 true somatic variants in this particular pooled material as well. But it's certainly not as big, obviously, as the germline set. So, the idea would be either to adapt the pipeline to still report germline variants, maybe report it in a separate file so you can use that for your analysis, or just examine the somatic side, in which case you'd have to do some analysis to, and well, here's the other thing, the positive set will be published. So you can always compare your positive set with your germline reference set. And so you know which ones, given the positive set, may be excluded if you do have a pipeline that excludes germline variants. And then you can just look at the remainder because those are the ones that should still be found even if you don't do anything to your pipeline. So there are ways around the issue of pipelines pre-filtering germline variants, but it is a good point. Since the majority of the variants in this sample A will be germline, you either need to adjust your pipeline to still report them, to be able to get the full use, or you can use the list of positives that we've identified and match that with your reference set so that you can see how many are remaining. And I think typically there will still be several thousand that you can use that are true somatic variants.
Along this line, that I was also wondering that what if that some pipeline report a false positive at the true negative position and happen to be look like a germline variant. And then of course, if a pipeline has such a filter, that would reduce the false positive rate. And so how would you think of that approach?
Yeah, so I think it really is a question of what the intent of the pipeline is. So some pipelines are trying to find all variants, germline or not, and even in cancer sometimes. For example, we just went over P53 in detail related to sample A. In terms of understanding the origin and the nature of the cancer, knowing whether or not a person has, say, one of the alleles for P53 may already be mutated, or BRCA1 and BRCA2, another good example of there may be predisposition to cancer because someone has a germline mutation in some key genes. So there definitely will be situations where there'll be contexts where pipelines will want to report even germline variants because they have relevance in cancer. It's just that, for example, if you were trying to validate a pipeline for tumor mutational burden, that would be a situation where you'd only want to report, somatic, so-called somatic changes. So again, I think the prescription for whether or not a particular variant is filtered or not and how to interpret that in terms of the performance of your panel very much depends upon what the intended use of the panel is and how you interpret the data from it and whatever parameter settings that you have with your pipeline. So I think each of these things are addressable. It's just that I think the solution is going to be specific to the intent of the pipeline.
Wonderful. Thank you for sharing your perspective. And thank you for the wonderful presentation, and thank you for the question and discussion. So for the rest of like 3 minutes, and then I would like to take that advantage and to remind you that for those who have registered for the webinar, and then obviously that you probably have also registered for the MAQC Society membership. We would like to remind you and to register for the MAQC Society meeting next month. So at the Society meeting, beyond SEQC 2, all these presentations, and then we're also going to have some keynote speakers, and then also have other areas of research that beyond the sequencing, and so we would like to invite you to the meeting and also to share your input, and then the society would come together and looking forward to the next phase of reproducibility study. So I just want to remind you of register for the society meeting. Wendell, would you also take a little bit time to introduce the next 2 webinars?
Yeah. So for the next 2 webinars, Dr. Binxing Gong next week will be discussing the tumor panel study, where we're using this reference sample primarily to interrogate, I think, maybe up to 8 different Oncopanels. Some of them you'll readily recognize. Some are, I think, still in development, and really examine, how do different panels perform. There's some very large panels. Some are trying to estimate mutational burden. How effective do we think they will be, in doing that estimation? Is there ways that we can improve estimates of mutational burden, because it is an important immuno-oncology biomarker. And then following that, Dr. Joshua Zhu, who has introduced me earlier, is going to speak on the results of the liquid biopsy panel testing. That'll also be exciting because now we're talking about variants at very low frequency, having to use the admixture samples to do that, and how those different panels performed using this material. So again, very exciting to see those results over the next couple of weeks.
Great.
I was going to say, I think with that, I think we're out of time. I think it's the top of the hour. So, so thank you for coming. We hope to see you next week as well, when we will continue this discussion on Oncopanels and their performance. And until then, thank you for your interest